All right. Shall we start? Okay. So yeah, today is going to be a bit of a not scattered, but we're going to touch a bit superficially on a few different topics, all related to, of course, the establishment of these cell compartments. We're going to finish or pick up where we left off the other day. So actually, yeah, sorry. This is a lecture I was going to give on Monday. So for a while now, we are just pushing back everything one lecture. And I guess we're still working on whether we're going to have some way to find a time slot for another lecture to have the full set, or if we're going to somehow skip and provide a recording from previous years. We're still working on this. Okay, so we're going to finish talking about the autophagy, the endocytosis, which is intimately connected to the secretory pathway we've been very busy with me. And then we're going to discuss somewhat briefly both nuclear transport, so in and out of the nucleus. Of course, it's a very important kind of process, and I'm going to make sure you stay awake by asking you questions. Right, that's helpful. Yeah, otherwise, you cannot do it. And finally, we're going to touch upon peroxisomes, which matter a lot. Okay, so we saw these slides before. Just a quick reminder that there are multiple entry points into the cells, and which route is being taken by the different components depends on their size, as well as some other things. We discussed this endocytosis, the receptor-mediated endocytosis. So remember some of the proteins that are really important for this? How is the coat made? Clathrin, right? So when you hear the word clathrin, you should see this movie of the triskelion coming together, forming the pit, and then the vesicle, right? Does clathrin interact directly with the cargo? No. There's the AP complex, right? And does that matter to be able to interact with the cargo? Yes. So, and interacts with what part of directly with the cargo? No. There's a need for a receptor. Yeah, I can't hear what you say, but I will surmise you mumble the correct answer. So indeed, right, just to make you guys think again about this, receptor-mediated endocytosis, creation of these vesicles once. So what triggers the coat to fall off again? ARF, GTP, yeah, GTP hydrolysis by the small GTPase ARFs, right? What triggers, oh, I forgot, I wanted to show you this. So if you go on Twitter, sorry, it's a tangent. I told you about dynamite, right? And I told some people yesterday in office hours about that. So I just saw 2 days ago a new really cool movie on Twitter from a modeling. So what these guys did is make a computational model of the membrane, especially when the vesicle is forming and is about to pinch off. And the model is about dynamine molecules. And the way, so I showed you these cartoons and these EMs, and I have not really showed you. Can I get less volume here? I get a lot of feedback. I love to do that. It feels like a rock star. You get more on the guitar. I made that joke before, so you don't have to laugh. So anyway, the dynamine. So the whole idea is that dynamine forms this polymer, right? And I showed you in the lecture, it polymerizes, and when you block GTP hydrolysis, polymerization goes in excess, and you get this really long neck, okay? But it never squeezes. And so what this model clearly shows is that if you imagine the neck of the vesicle, dynamine forms kind of an ellipse, or a helix, sorry, I should say, around that neck. And that helix forms by polymerization of dynamine monomers, right? But then, like I told you, dynamine is a molecular motor, and then what happens is upon GTP hydrolysis, not only is polymerization stopped, but also the necks kind of squeeze it. So you can very easily imagine if you have an helix, it's very easy to squeeze it this way. And that's how you have the force that's exerted by harnessing the energy of GTP that's converted into squeezing that neck. Anyway, so back to this. Right, we talked about how these vesicles, once they shed the coat, are now capable of fusing with the late endosomes, and it's only upon fusion with the primary lysosome. So now this is material that comes from the secretory pathway. So I'm not going to do a whole review session because we have one tonight, but that should trigger memories about the M6P and the lysosomal enzymes and the receptor and etc. Okay. We're not going to talk much about phagocytosis, but those phagosomes, which specialize into engulfing larger particles like bacteria, also have to fuse with these primary lysosomes to make secondary lysosomes. And eventually, so we're going to talk about what happens here. But the end goal of this is to break those complex micromolecules into smaller components and recover them. So amino acids and different kinds of lipids, be they cholesterol or fatty acids. Okay? And I also showed you this next slide the other day. And so we're going to talk about different aspects here. So like some of the electron microscopy images I show you, one reason is to make you appreciate that data, this is kind of raw data, is often meaningful once you've understood what happens, right? And so in this case, both these vesicles here and those ones are so-called autophagic vesicles. This is kind of an advanced one where the elements have been digested to some extent. And this is a newly formed one, and I'm going to show this again and tell you more. This is a mitochondrion, this is a peroxisome. So those organelles can be digested. And I want to pause for a second here to say it's very important for the cell, right? So in this course and in my lectures, we've talked a lot about nitty-gritty and all kinds of details. But if you step back and think about cell physiology, sometimes they starve. And then they alternate and they are all of a sudden, well nourished and they have to do a lot of work, right? So these organelles, the total amount of mitochondria, of peroxisome, and so on, varies. The cell is constantly kind of monitoring its own state and eventually remodeling the composition of its cytosol and its cytoplasm. And part of it means when there's less energy and so on, or some particular signals, mitochondria, peroxisomes have to be digested, have to be degraded, right? If you remember the lecture on mitochondria, it's very expensive to keep mitochondria going, okay? That's just to make you connect the dots here. But I'm going to start by telling you a bit more about this one here. It's the so-called late endosome, that's what this acronym stands for. And one of the things I really wanted to highlight is that most of the lumen contains this really electron-dense material. So what do you think is this density due to? We've seen this kind of stuff before. Protein aggregates, right? Protein aggregates, what could be contributing to aggregation of proteins? Low pH, right? Good. That's indeed a big part of it. And these enzymes, many of them work, be they the peptidases or the lipases at low pH. But you also see these structures here. And if you take a close look, it's not easy to see on this one, but they are membrane bound, right? So it's kind of peculiar because you have a compartment, this late endosome, and there's a compartment in the compartment. It's like Russian doll compartments. So it's also called a multivesicular body. And we're going to see that it's very important to achieve to solve one of the conundrums or one of the problems that lysosomes have to solve, which is how do they know what to digest and what not to digest? Okay, so we've seen the case of the lysosomal enzymes. They have these particular glycosidic modifications and so on, and they are part of the process. So somehow the lysosome knows those are lysosomal enzymes. But for membrane proteins, it's not so trivial. Okay, and we'll see that these intralysosomal vesicles are part of the solution. Okay, so if we go back to the kind of the dynamics of the system, here it goes. So we saw, and I showed you this slide before, you have to form the late endosome where things are starting to happen. You have the merging of vesicles from two different routes. One of them, in this case, endocytosis. We've seen this almost ad nauseam by now. And then this other route, that's where the enzymes come from. That would be the primary lysosome, if you will. And once they fuse, now you have a mixture in the same membrane of membrane proteins that matter for the function of the lysosome. So, for example, these green bars here, and the blue bars. It's just a representation, right? Okay. You know that. And so one question is, how can this lysosome know that the blue bars have to be digested, but the green bars have to be kept because they matter for, say, lysosomal function? Okay? And that's where those vesicles kick in. So one idea, it's very, I don't want to say clever, it's a very elegant mechanism here, whereby you actually create different membrane-bound compartments. And what's depicted here is that you're sorting, basically the green bars, the lysosomal membrane proteins, remain on the outside, but then the proteins that are destined for digestion and degradation, instead, they make it into this intra-lysosomal or intra-endosomal vesicles, and later on, they are the ones to be digested. Okay? So something really essential happens to distinguish proteins that have to be sent for degradation from proteins that have to be kept at the lysosomal membrane and protected from digestion. Okay? So here it goes. Okay, so the first thing is, and I'm just going to give you a couple, detailed insights into this, and then one interesting way a particular virus hijacked this machinery. So the machinery we're talking about starts with this HRS protein here, which helps monoubiquitinylate other proteins. So what that means, so you all know what ubiquitin is? Yeah, small protein. That's often used as a tag. It's added covalently to other proteins. What's a very classic example of ubiquitinylation?
Tagging for degradation.
For degradation, right? So it's polyubiquitinylation. So you have many ubiquitin proteins being added in a very specific way to target proteins. And all of this is extremely specific, right? So I'm not giving you many details, but one idea is that this HRS protein acts as a ubiquitin ligase or part of a ubiquitin ligase complex and helps ubiquitinylate these cargo proteins or proteins that are fated for to be degraded. Okay? And that monoubiquitin tag is essentially the tag that's starting to tell the system, put this protein into a vesicle. Okay? So that's a first sort. Now, that doesn't tell us how the vesicle is formed and how it breaks off from the lysosomal enzyme. So that's where this other complex kicks in. So it's called ESCRT for endosomal sorting complexes required for transport. I'll forgive you if you don't memorize this, so don't worry. But the idea is that it assembles, so it forms a polymer. This assembly is triggered by the presence of this HRS protein, right? So this HRS protein really works like a signal. So if it helps you remember this, it's kind of analogous to, say, the SAW protein helping recruit the coat that has a bit of a mechanical role to play, right? In this regard, this is as far as this analogy goes, right? These are very different proteins and it's a different process. But you get the idea. You have a signal, you have a trigger, and then you have some machinery that helps achieve the formation of these vesicles, and now you have a vesicle. So sure enough, this complex exerts force, it helps form the vesicle, it uses ATP, and so on. Okay, I'm not giving you a lot more details than that. Well, sorry. What uses ATP really is the recycling of this vesicle, of this complex. And that's, I should say, the analogy here is more with, actually with which other complex? Which other complex forms helping membrane remodeling and then has to be deassembled for recycling, and that deassembly needs ATP because it's such a stable complex. The snare complex, the cis snare complex. And why do we need to deassemble the snare complex?
To recycle.
To recycle the V snares. Right, very good. Okay, so that's all I'm going to tell you about this. Okay, yes?
Where does the HRS protein come from?
That's a good question. The secretory pathway? Where else? Yeah, no, I mean, that's a good question. Yeah, it's probably assembled on the primary lysosome as they go. But I would have to double check to tell you for sure.
So is it assembled near the cargo protein?
Is it assembled near the cargo proteins? Well, okay, so in this cartoon, I would say the HRS comes with this.
Okay.
So it's part of the lysosomal machinery. So it eventually meets the cargo protein, but it can't meet it from the get-go. Does that answer your question?
Sure.
In other words, they can only meet here.
Okay.
Now, the reason why I have a slight doubt is that this protein does not seem to be a transmembrane protein. So for those, sometimes they can associate with the membrane secondarily. In other words, bypass the whole secretory pathway if they are tied by acylation, for example, or farnesylation. Okay, so it may be one of those. That's the stuff I would need to double check. See, I'm not making things up. When I don't know, I'm telling you, I don't know. I know it's disappointing. You guys thought I knew everything, but I don't. Sorry to disappoint. I just know more than you guys. Okay, never mind. So there's one particular thing with this ESCRT complex. It's of course very important for the cells, and it's been shown to be hijacked by a virus and the HIV itself, human immunodeficiency virus. So, and the hijacking goes exactly by targeting what HRS1 normally does, which is, that's also, it's related to your question because in this case, it doesn't come from the secretory pathway. It's actually added onto the membrane from the cytosol. So it's the HIV gag protein. And what this gag protein does is very similar to HRS1, right? So if you look at the previous slide, HRS protein is here, helps tagging proteins and recruiting the ESCRT complex. So this recruitment of the complex is essential to make the vesicle. And the viral protein does exactly that, except it's at the plasma membrane. And it's triggering also the formation of now what you could call an exosome, or a vesicle that forms towards the outside of the cell. To do that, it does recruit the same ESCRT complex here. And that vesicle is essential for the virus because this is where the viral particle, this is how the viral particle is formed, right? So, sorry. Right, so at the end of the day, it's the same machinery, ESCRT complex, leads to the pinching off of that exosome, that viral particle, and now the virus can go. Right, so of course this has been studied extensively, and if you block this TSG101 protein, it's a protein of the host, right? It's not a protein of the virus. Okay, that's the principle of the virus. It has just a few proteins that help him or help it hijack the host machinery. Okay, so in this case, this TSG101 is one of the proteins involved in recruiting the CRT complex. Yes.
That was a host protein from TSG1?
Yes. So exactly. So TSG101 is this protein. It's part of the ESCRT complex, and that's the one that connects with the HRS in the normal case, or with the viral gag protein here. Right. So all the virus had to provide here is the gag protein. It substitutes for HRS, except it's at the plasma membrane, and what's being packaged are not enzymes or protein bound to be degraded. Instead, it's the virus itself, right, with the genomic material and so on. And it also helps recruit the host machinery, the ESCRT. And then if you block, that's what this image is showing. If you block TSG1, so you use a TSG1 mutant host, the virus, you see the viral particles can begin to form. Here they can detach in a wild type, but eventually they do not detach. Right? So what this is illustrating is that it's a host protein that's necessary for completion of the formation of the viral particles. And that's the exact same process. So you would say, well, but that host cell must have also problems with its own multivesicular bodies, and sure enough, they do. Okay. All right, so to finish quickly with this, it's the same cycle all along. You need to recycle this ESCRT complex, and it also uses the energy of ATP and so on. All right, so that's all I wanted to tell you about the multivesicular body and what happens in endosomes. I told you today we're just going to sample a couple different stories, a bit superficially. So now let's talk about the last part, promised this autophagy. Right, so in cartoon forms, I already showed you everything winds up being digested in the same way in a secondary lysosome using lysosomal enzymes. So those, right, again, those proteases, those lipases, they have very little specificity. Okay, you cannot expect them to see a protein and not digest it because it recognizes it as an important one. That sorting is done independently, like I just showed you. But it starts in a different way when it's about autophagy, and we'll see it involves the ER and the formation of these extra vesicles. So if you take a closer look at these autophagic features here, especially in an area like this, you have the two membranes of the mitochondrion. And here you see they are surrounded by not one, but two membranes. Okay? So you basically have a double membrane closed compartments which encases those organelles. By the way, do you know anything about these two mitochondrial membranes? The inner and the outer? Oh yeah, that's on the exam, huh? I know, I can almost see all the flashes you all have now with the redox chain and the electron transport and the electrochemical gradient and so on. Anyway, I'm sorry, I'm kind of longing for the times when I was teaching that part and Carlos. Anyway, so here it goes. I mean, it's pretty straightforward, quite frankly. So you have two kinds of autophagy. You have one that is here annotated as random initiation, and there was actually a Nobel Prize attributed to the discovery of autophagy a couple of years ago. And one of the work was on this protein ATG8, which is one of the early determinants of the autophagic process. And some of them are specific, so there's a signal to degrade specifically either peroxisome or mitochondria. So to have a targeted autophagy of special organelles. Okay, it's one of the main differences. But in both cases, it starts in the same way. You have this so-called autophagic vesicle nucleation, and it's quite simple. You just imagine if you have some 3D view capability that these two membrane compartments actually a leaflet, okay, of the ER. So if you remodel that, you can very easily encase anything, okay? And indeed, this is what happens. So that's why you have now these two membranes surrounding an entire organelle. But now you've basically created this autophagic vesicle where you have the organelle encased in a particular compartment. So all you need to do next is to have this autophagic vesicle fuse with a lysosome. Okay? And then it's going to resemble what you get from a multivesicular body using the ESCRT complex, where you have the lysosome. So everything that's on the outer membrane is somewhat protected against digestion, but everything that's inside is fated to be digested. Right? So that includes this membrane, that includes the organelle, and so on. And of course, ultimately, so here it just says amino acids, but you could imagine it also includes sugars, lipids, and so on. Then the cell needs to recover those components. And so the lysosomes, they also have these important amino acid permeases, right? So just to illustrate, because I'm telling you, there are some important membrane proteins on the lysosome, but you should be skeptical. You're a scientist in the making. So you could be like, okay, what kind of membranes? Membrane proteins. These kinds of membrane proteins. Okay? What other kinds of transporter are there at the membrane of lysosomes that you think would be very important? Which I already told you about, and also are on the exam. You didn't see that one coming, did you? So, what's a particular property of the lumen of the lysosome? Acidic. How come? Proton pump. You want to degrade your proton pumps? No. So that's one example, right? So you see, you have to connect the dots. Anyway, so I'm just trying to spice this lecture up because it's a little... Okay, so let's talk about something else now. We're going to transition, almost switching gears, and now we're going to start talking about the nucleus. So lots of transport has to happen in and out of the nucleus. So can you think of a couple examples of non-trivial transport? So macromolecules, mRNAs, very good. Where are they made? In the nucleus. What do they have? Where do they have to go? In the cytosol, the cytoplasm. Why? Because that's where they are translated. That's exactly right. Ribosomes, they are made, you know that? You know where ribosomes are made? In the nucleus. You know a lot, actually. And they also have to be transported. Those are big macromolecules, macromolecular complexes. These transports are not trivial. And we're only talking now about transports from inside the nucleus to outside. Now, are there transports, can you think of transports needed to bring things inside the nucleus? Polymerase, that's a good one. Helicase, like all kinds of proteins actually. And why do we have to bring them inside the nucleus? Yeah, that's the function, but why not make them in the nucleus?
Oh, there's no ribosomes in the nucleus.
That's right, because translation happens in the cytosol. I know, I give it to you. It's obvious stuff. But sometimes it's kind of a feel-good question, so you know the answer, and you're like, oh, I'm so happy to be here. Okay, so nuclear import. So very much like some other kinds of transport, the information, the zip code, in many cases, is in the protein. But, and I wanted to highlight that on this slide, we're now entering the realm of decoupled translation from transport, right? So I've insisted a lot on this co-translation or translocation. Now for all the transports we're going to talk about today and on Monday, this is translation first, then there's a targeting sequence, that tells the cellular machinery where the protein has to go. And those are the things we're going to talk about. Okay? So in this case, proteins can go in fully formed, fully folded. And that's important because that means in some cases, this is going to be a bulky structure. Right? So this is also by contrast. So the contrast might help you remember this with, of course, the translocation of the proteins in the ER, where it's completely unfolded. You basically, it's a string of amino acids, and the folding happens after translocation. We're going to see next week, transport to the mitochondria and transport to the chloroplast also involves unfolded proteins, and the folding happens after translocation. In the nucleus, it's different. The folding comes first, and then it's a fully formed protein that goes in. Now, one thing you can already anticipate when I tell you this, and that's why I'm insisting on this, it's because the transporter is going to be different, right? Think about it. The system has to transport a fully folded, big, bulky molecule. That means you can't do it through the tiny hole of a translocon. You do it through a big nuclear pore, right? So I'm going to show you this. That's why these two features are essentially tightly coupled. Okay. Now, a lot of these transport are also polarized. You actively transport proteins either from the cytosol to the nucleus or vice versa. So we're going to see how this is achieved. And the localization sequences, they can be nuclear localization sequence, or I don't have it here, but nuclear export sequence. And as usual, they are short. There's lots of variability, although there are softwares to recognize them because they have patterns in common, and so on. So the nuclear pores. So those micrographs are showing you, I don't have a scale here, which is too bad, because these are really big macromolecular complexes. They are very stable. So it's thought that the progressive degradation of nuclear proteins is part of the aging process, because unlike mitochondria, unlike some other organelles, some other complexes, those are not renewed as often. So they survive through mitosis even. They just reassemble with the nuclear envelope after mitosis. But what I want to illustrate here is the polarity. I mean, there are two things. So if you look at this, and if you think about what am I seeing, and how would I describe it? One thing is whether you watch it from the cytoplasm or from inside the nucleus, or nucleoplasm as it's sometimes called, those nuclear pores look very different. Okay, so there's a marked polarity, cytoplasm to nucleus, which you can imagine will have a role to play in the directionality of the transport. Okay, there's another feature that's quite obvious when we look here on either side. This one, so this one is a bit broken, or this guy here. So it's complex, but there's a symmetry, a so-called radial symmetry. So one idea is that the nuclear pore itself is made of dozens of proteins, but some of these proteins are repeated multiple times and contribute to the symmetry. And the last point I'm going to make is, how many do you see? Many, right? So it's a trivial point, but the idea is that it's not like there's the nucleus and there's one door, right? There are nuclear pores all around. So this is a testimony, if you will, to the intensity of the transport in and out. Things can go in and go out of the nucleus pretty much everywhere. Okay, so in cartoon form, this is how it looks. So here you see the cytosolic side, these so-called cytoplasmic filaments, the basket, terminal ring, etc., etc. Big macromolecular complex. Quite frankly, do I need you to remember a lot of details about this? Not really, right? Just to be able to think about the concepts. And the one thing for which I want to that I want to illustrate with this particular image here is the nuclear envelope. A lot of people, including colleagues, faculty members, researchers, etc., talk about the nuclear membrane. Don't talk to me about the nuclear membrane. It's called the nuclear envelope when you talk about the whole thing, because these are actually two membranes. There's an inner and an outer membrane, but they form somewhat of a continuum, right? So it might be a bit misleading to call them two different membranes, outer and inner, although they have different properties, they have different compositions. This one's, the inner nuclear membrane interact with the nuclear lamina here. And those compartments are very important for gene regulation. Did you talk about that? Did you hear about that in MCBII at all? The nuclear compartment near the lamina and the role in gene silencing or gene activation and so on. You talked about this a little bit. Okay, so this is connected to this, right? The structure of the nuclear envelope. And the outer membrane, and I don't have anything about this on this course actually, but that's too bad, because there's an emerging field whereby you have proteins at the outer nuclear membrane, which are somewhat connected to what happens outside the cell, such that the cell, and on the other hand, connected to what happens inside the nucleus. So those are important to have a so-called how mechanics or physical perturbation, let's say you poke a cell, will have an impact on gene expression, right? So it's not trivial, it's not the classic signaling pathway kind of thing, but there's a way the cell can sense the mechanical environment and respond by changing, fine-tuning gene expression that involves, of course, proteins at the nuclear envelope. The last point I'm going to make about the nuclear envelope is that, so you see, you have essentially a continuum, and that's where the pore is. So strictly speaking, when proteins or macromolecules are imported inside or outside the nucleus, they never really have to cross a membrane. Right? They kind of go around using these dedicated gates, if you will, the nuclear pores. Okay, so let me show you now a couple simple experiments to study these NLSs, or nuclear localization signal, and so on. So let's start with this simple protein, pyruvate kinase. What is that? Have you heard about pyruvate kinase? Yeah.
The phosphorylate pyruvate.
It phosphorylates pyruvate. Good call. What kind of process is that about? Glycolysis. It's the metabolism, right? Where do you expect it in the cell? In the cytoplasm. Glycolysis is in the cytoplasm. Watch out. This is coming up Friday. It's in the cytoplasm. So normally, the whole point is normally that protein does not go into the nucleus. Okay? And one reason why it doesn't go is because proteins above a certain size, they can't diffuse passively through the nuclear pore. Small proteins can. It's big enough. But big proteins have to be specifically targeted and transported, actively transported into the nucleus to make it there. And you can force them to do that. So if you take pyruvate kinase and you add through molecular biology techniques and cloning and so on, you add a tag, a so-called nuclear localization signal to this pyruvate kinase, which could be in this form. So they usually are rich in lysines for some reason. Now, most of it winds up in the nucleus. They tend to remain kind of excluded from the nucleolus, which is a special nuclear subcompartment, but they really are transported, right, actively into the nucleus. Okay, so this is a good assay. Remember, I told you, when you do experimental biology, you always need at least two things. You need an assay, something you're looking at, you're measuring, etc. So here we can be measuring the transport of this particular protein into the nucleus. Okay, and we know from this experiment it depends on the presence of this NLS. If you did the opposite experiment, which is you take a normally nuclear protein And you study the sequence and you recognize there's a small stretch of lysine-rich amino acids and arginine-rich which resembles an NLS. And you mutate this or you remove it. Now that protein is unlikely, if it's big enough, it's probably not going to make it in the nucleus. Okay, so these sequences are usually necessary, that's what's showed here, but also that's sufficient, and it's also necessary in the experiment I just alluded to. Okay, so having this assay, now we can study the machinery, right? In other words, what is needed to have this transport happening. And for that, we need perturbations, right? So one of the perturbations I'm going to show you is the following. And this is an experiment that every year tends to confuse people a little bit, so pay close attention. So the idea is to take cells in culture in this experiment and to treat them with this component digitonin. So what digitonin is a light detergent. In other words, it's going to solubilize membrane lipids, but it's not going to break down the membrane. It's not like SDS or one of these strong ionic detergents. It's very light. Okay, so only a fraction of the lipids is going to go away, enough to permeabilize the membranes. Okay, but not enough to disrupt the cell. So that's why the cells still look pretty good, but some stuff is gone. So for example, if you look here, you see inside the nucleus, it turns out some material which was a bit more dense on the control side it's more clear here. And so the idea is that now it's like you have a ghost of a cell. So the nuclear pores are still in place. A lot of the protein, the complex protein structures are still in place, but the soluble stuff is gone. Okay, that's the important thing to understand with this premise. And then the experiment is the following, is to then add back some lysate. Okay, so imagine you prepare your cells here. You have your protein you're going to look at and ask, does it make it into the nucleus or not? And you have some lysate from cell that you're going to test for its ability to rescue. So when I say rescue, here is what I mean. So this is just a black square. I'm sorry. But what the black square illustrates is that it's not like this one. In other words, if you try to monitor a protein inside the nucleus, you don't see anything. And that's because, sorry, just to be clear, right? Now we're looking at digitonin-treated cells. Okay? So in digitonin-treated cells, some of these soluble proteins which normally make it into the nucleus do not make it into the nucleus. That's the black square. But if you add back some lysate, you can have that protein go into the nucleus. So now you have a positive control, and so you can imagine that you can now fractionate using biochemical methods the assay and add back different fractions and ask what is needed for the transport. Okay? Very tedious biochemical work, but that was part of what led to the models of the nuclear import-export cycle I'm about to tell you. Yeah?
So in order to see, you need the digitonin and you need the lysate? You need both.
Sorry, can you say that again?
So in order to see, you need both the digitonin and the lysate?
Yeah, so the digitonin is without digitonin, the protein makes it into the nucleus because there is everything the cell needs. So the digitonin is a way to remove the soluble component from the cell and just keep the structure, the nuclear pore, and so on. Okay? So the nuclear pore acts like a gate, but for big proteins, it lets them through only if they have the right tag and if there's the right soluble components, which I'm about to tell you. Okay? All right. So here's the cycle. So it starts in the cytosol. Why does it start in the cytosol? For obvious reasons, that's where the protein is made. It's fully folded, but it always exposes the NLS. And in some cases, actually, if you ever wind up in a lab and you're asked to create a chimeric protein with an NLS and you want it to work, sometimes it won't work if for some reason when the protein folds, the NLS is inside, right? So it's always kind of sticking out. And it sticks out such that it can interact with one protein here in the cytosol called importin. And here it's drawn in a way showing a conformation that's compatible with interaction with this cargo protein. And here, this black box here, that would be the NLS. Okay, so very simple. You have a signal, you have a receptor, the importin. When this is, so this conformation here is because it's bound to a cargo, so it's a conformation that promotes transport through the nucleopore into the cytosol. But now we have to do two things. One is release the cargo into the nucleoplasm. The other one, as often, is recycle the importin. Right? So you can already imagine having, gone through all the transport in the secretory pathway, that the importins are going to essentially shuttle in and out of the nucleus. Okay? So indeed, this is what happens, and it involves now a new family of small GTPases, and that should now ring a bell, which come in two flavors, GTP bound or GDP bound. Which one you think is going to be the active form? The GTP bound, right? It's a quasi-universal truth. And the main difference is, so GTP bound, there's one conformation here, GDP bound, there's another conformation. So what happens is you have the GTP bound form in the nucleoplasm, and it can basically compete with the cargo for attention of the exportin. Right? So it binds the exportin, and it's a real competitive binding. It changes the conformation of the exportin. And now you have this complex. So the cargo is released in the cytosol. And now this complex, the conformation has changed, shuttles back into the cytoplasm. Right? So we are close to having now recycled the importin. And indeed, here there's hydrolysis of GTP. So now the RAN can let go of the importin and we've fully recycled. But now we have to recycle something else, and that's the RAN. The GDP bound form. And it just comes back here. But the last point I'm going to make is not only do these proteins shuttle in and out of the nucleus, they also have to shuttle between GTP bound and GDP bound. And for this to work, you may appreciate that the GTP bound form must be on the nuclear side, the GDP bound form on the cytosolic side. Okay? So how you're going to regulate that? By positioning the GEF and the GAP in the corresponding way. Right? So that's why the GEF, so the exchange, so the reactivation of the RAN, if you will, happens only on the nuclear side, and the GAP on the cytosolic side. Okay? So it's the polarity of the GEF and the GAP that determines the polarity of the whole system, if you will. Yeah.
What changes the conformation of the importin? Because it looks like in the diagram it changes, like the red one.
Between which step and which step?
When you go from like over there to
from here to that?
Yeah.
It's the competition, is the fact that the gap, the RAN, GTP here, will basically force the change of conformation in the interaction.
Okay.
That, yeah.
Yeah.
It's protein-protein interactions. And so you basically what you can take home is that the affinity of these proteins. So the cartoon, I see what you mean. The cartoon is a bit misleading because it looks like what comes first. It can't bind if it doesn't change conformation, etc. Well, the molecular reality is a bit more complex than this, but that's to make the point.
Yeah.
Okay. But yeah, it's more affinity, competitive affinity. Okay, so okay, now let me ask you one question. What do you think would be so the GEF and the GAPs, these are other proteins. We don't need to look into the details, but where would you want to put them? Well, on either side, right? One we want it on the cytosolic side, one we want it on the nuclear side. But can you think of a polarized structure that's different on the cytoplasmic side from the nuclear side, that's close to where the transport happens? The nuclear pore itself. Right? I showed you it's a big complex with this basket on one side, so it's big and polarized. So if you associate the GEF with this nuclear side and the gap with this cytoplasmic side, you basically build in the polarity. Okay, and that's one. So now you have. I showed you the structural polarity of the nuclear pore. Here you have a functional polarity. You have a reason to polarize it, if you will, to polarize the transport of these proteins. Now we saw that other proteins have to go the other way. Okay, for that they have a nuclear export signal. So it works. It uses the same machinery essentially, but in different way. So the export cycle uses nuclear export signals, and the key difference is now the presence of this nuclear export signal here is this exportin. And so here how it works. Now exportin cooperates with RAN-GTP for binding to the cargo. And again, now the binding has to happen on the nuclear side. Altogether, and it's GTP bound because that's the same RAN. So the GEF, it's the same GEF, it's the same gap. The key difference is the NES and the exportin, right? They basically just use the GEF and the gap differently. So indeed, on their way out, they come together at a trimeric complex through the pore. Now, on the other side, they meet a gap, which promotes hydrolysis of GTP by the RAN. That causes the dissociation of the whole complex. And then both the exportin and the RAN-GTP can come back through the nuclear pore. So you may ask, well, don't they need their own NES or NLS to go in and out? No, because they are small enough. Okay? So these are really small proteins, so they can basically diffuse back into the nucleus. All right, briefly, RNAs. I mean, we could have almost an entire course on RNA localization, RNA transport. We're going to have one slide for the moment. Right, so there are just a couple key concepts I want to convey with this slide. One of them is that RNAs are never alone in cells. They are always associated with proteins forming these ribonucleoprotein complexes or RNPs. And the kinds of proteins that associate with newly formed RNAs in the nucleus have these NXT1 and TAP1 complex, which are the export proteins themselves, and these DBP5 protein, which is a helicase. So first concept I want to drive home is always associated with proteins, right? So in other words, transporting RNAs boils down to transporting proteins. And then you have the RNA almost. And the specificity of binding RNAs is with the protein themselves. Then the other point is this DBP5 is a helicase. So what helicases do, you've seen some of them in molecular cell 1, they can unwind nucleic acids. And for that, they use the energy of ATP. But they also kind of move along nucleic acid. So there are two functions for this DBP5, which is, so unwinding, so RNA can become really big and bulky. So normally that's not a huge problem for nuclear pore. They can accommodate that. But there might be some roles for unwinding, the more tangled parts of the RNA. And the other angle is you have DBP5 associated with the outside of the pore, which may help pull the RNA out, right? So bottom line, the helicase activity may be involved in both the conformation of the RNA and the active movement of the RNA, right? So these are the two main points I wanted to drive home. And then once the expert is done, the RNP remodels, so now the RNA is licensed to go and be translated if it codes for protein, while the other transporters shuttle back. All right, so that's what I'm going to say about the nucleus, and I'm going to not going to lose the last 15 minutes. I'm going to use the last 15 minutes to tell you about peroxisms. Any questions on the nuclear transport? All right, let's do this then. Okay, so peroxisms. They are tiny organelles, quite simple, single membrane. They don't have their own DNA, unlike mitochondria and plastids. And they do a very important job of regulating the oxidation or oxidative status in the cell. So there's this reaction here, which happens primarily in peroxisms, and that's to, if you will, detoxify. Well, sorry. So these kinds of reaction happens a lot in cells. What is specific to peroxisms is the handling of H2O2, right? So that would be very damaging for cells to have too much H2O2 in them. And so there's this enzyme catalase which helps degrade H2O2 into back into molecular oxygen and water. And so in a way that's the main job of peroxisms. So it's an important job for cell integrity. So there are two components that we need to think about is how do you create this compartment, right? So in other words, you're going to have a compartment where the oxidative potential is really high, is higher than most other places in the cell want. Okay, that's the peroxisome. That's where this oxidative potential is handled. So you want to create the boundary. So you want to create that membrane, and then second, you need to load it with catalase, right? So in terms of transport, these are the two kinds of transport that are needed. Transporting proteins to the membrane, transporting catalase into the peroxisomes. And those peroxisomal targeting sequences have begun to be characterized. They are quite simple, like this C-terminal 1 SKL. So it's a C-terminal tag. So again, it's consistent with this idea that the transport is decoupled from the synthesis, right? Synthesis happens in the cytosol, fully formed proteins show up, and then they are being transported. So let's just walk through some of the mechanism. So PTS1 appears on this peroxisomal targeting sequence, and it's recognized by a diffusible receptor, which can basically snatch it wherever it is in the cytosol. Does that remind you of something? A diffusible receptor that can recognize a free-floating tag in the cytosol. SRP, that's how SRP works, right? So it's analogous here. The PTS1 shows up, PEX5 recognizes it and helps bring it to a receptor on the peroxisomal membrane. And then there's formation of this complex, which is the transporter itself, right? So again, this is quite analogous to what we saw, SRP, the SRP receptor, the Sex61 translocons, okay? And then the peroxisomal protein can wind up in the matrix. That would be, for example, catalase, right? Catalase is a soluble protein. It's in the matrix of the peroxisome. The trick here is that, PEX5 gets in, so this is peculiar or special for this system, it is transported all together, right? So it basically takes the matrix protein all the way into the matrix, and then itself it's exported back into the cytosol. That's how the recycling happens. It doesn't let go here like SRP does. But the part I kind of enjoy about this peroxisome. You wrote that, right? When I say my favorite part. It's not so much this, because this is very simple. I like more subtle stuff. So the experiment itself is quite elegant. How you distinguish between, how do you build a hierarchy of transporter for the membrane and the matrix? And connecting back to the first part of the lecture, these peroxisomes, they can be digested by the cell often, right? So the population of peroxisome in a cell is not a very stable thing. Peroxisome are made, peroxisome are degraded. So peroxisome biogenesis is a key driver of the number and of the amount of peroxisomes in a cell. So there's constant biogenesis, there's constant autophagy of peroxisomes, okay? So that's why this process of making them is very important. So here's the assay. So let's say you have your peroxisomes and you have two kinds of proteins that you can look at. One of them is this PMP70, so that's one of the transporters. And it's at the membrane. And then there is catalase itself. Some of it is in the cytoplasm. Of course, that's where it's synthesized. But then it tends to accumulate and be more concentrated, hence the brighter signal in these peroxisomes. And if you look closely, so this is the same cell, just two different labels. You have almost the same image that you can superimpose, which makes sense, right? Because at this magnification, you can't tell apart the membrane from the lumen. Peroxisomes are small. Both you see dots, okay? So, yeah, just to be clear, because this in the past has confused people, this would be a whole cell, right? Not the whole peroxisome. And so each bright signal here is a peroxisome. And you see in some cases, like this guy here, if I'm not mistaken, there's not so much catalase. So what do you think comes first? Imagine you have to build a peroxisome from scratch. What do you start with? PMP70 with the membrane. That's right. You have to start by making the compartment. And so indeed, when you have mutants that affect the first steps of the biogenesis of the peroxisome, let me take that back. That affect not the first step, but the later steps, the loading of catalase. You can get these kinds of pattern where when you look at PMP70, you see dots. So some peroxisomes are there, but none of them contains a high concentration of catalase. Instead, catalase seems to remain outside. So this is one evidence that this PEX12 gene is important for the transport. So those are called peroxisomal ghosts. And the model is that the peroxisomes exist, their membranes are there, PMP70 is there, but it's unable to transport catalase inside. And then in acute cases, like PEX3 mutants, you get nothing. What do you think happens in PEX3 mutants? Not even the membrane is made. That's right. Right, so that's the model. Indeed, the proteins are synthesized. Why can we synthesize the protein? Because if this was an ER protein, it could be problematic. Right? We talked about co-translation, translocation, but here, bear in mind, it's decoupled. So the synthesis happens no matter how. It's the transport that does not happen. So that's why you get some haze of a signal throughout the cytoplasm. Okay, so my final slide is just to recap how peroxisome biogenesis happens. There's a precursor membrane. So by and large, I don't know how much Carlos insisted on this, but a lot of the membrane biogenesis, like default membrane, happens at the ER, the smooth ER. So for peroxisomes, for example, for autophagosomes, I showed you need this membrane with ATG8. You tend to start, you kind of take membrane from the ER complex ER. So that's where the precursor membrane would come from. And then you start singling it out in some ways and adding this peroxisomal membrane protein, and there's some order, right? So in this order could be worked out in part through this kind of assay I showed you. So indeed, first, the membranes are made. That's how you get these peroxisomal gauze, but now you have all the machinery needed to start loading it where the catalase needed. All right, I stop here, and I'll see many of you tonight, I guess.